So yeah, I just want to start off by kind of acknowledging and introducing our team a little bit. I've known a lot of the folks on this webinar here for a number of years, so you've really seen kind of the development of Pipsqueak and the development of Rewire. And so we're at a pretty cool point where we have a lot of people who are putting in a lot of hard work on this. I think Christy and I, since we tend to be the ones talking in all these meetings, but we have a whole group of people behind us. Barb Sorg, I think, is probably someone who's familiar to a lot of you. She's the person that I did my postdoc with. And so we've got some great advisors, on our scientific board, but we have a ton of people who are developing, the really neat features in Pipsqueak now. With us today, we actually have Will O'Keefe and Grant Wade, who are two of our software magicians who really do some amazing work. So all of the custom AI stuff that we'll show you today really goes to these two putting it together. So pretty cool. And I think on the call, we also have Daniel Lockridge, who is brand new to our team and someone that you might get to interact with here in the future. He's coming on to really help us build out our products and our customer base. And so Danny is a great advocate for what we're doing and just a really affable person. So watch for emails and phone calls from him. But yeah, so Pipsqueak has come a really long way, and I like to acknowledge our 2016 paper because this is really where we first described kind of the rationale for what we're doing with Pipsqueak. Generally, we want to improve the accuracy and quality of microscopy image analysis. So in my postdoc, this meant that I was doing a lot of confocal image analysis and then trying to count or otherwise quantify the intensity of some cells like perineuronal nets or parvalbumin staining. What we found in my lab was that it was very hard for us to be accurate across individuals, and especially if we were to bring in summer interns or people like that who just don't have a lot of experience. And so we wanted a way to standardize it and kind of improve the accuracy. And so that's what we published in 2016. And since then, we've continued to build on this and evolve it. And where we are today is really incorporating artificial intelligence to kind of hit that last sticking point of detecting cells accurately. And so at this point, I'm sure you've all seen kind of our little blurb on the website and this cute little video. But the meat of what we're doing is really trying to train computers to do that tedious cell detection for us. And downstream from that, Pipsqueak hasn't changed a whole lot. What we want Pipsqueak to do for you is to quantify the pixels in an image that you show to it. And if it can help detect that cell more quickly and more accurately, then that's a great benefit on top of it. And so that's really where we see the value of Pipsqueak, is that this is improving the reproducibility of our data extremely significantly. It's also reducing the time it takes to go through an image. And I think most people who have gotten to use Pipsqueak at this point can probably attest to that it takes me about a minute to do an image in Pipsqueak, whereas it would take me 15 to 45 minutes to do by hand. So huge benefit there. And then that also just really reduces a lot of costs and pain points in our image analysis workflow. This is the last thing that I always did in an experiment, and so is the last thing that I wanted to focus on. So Pipsqueak takes care of that. And so, that's really led to this adoption around the world. And I can't thank our users enough for all of the citations. I think we're up to about 40 citations of Pipsqueak now. So you can actually find a publication or a list of publications on our website at rewireneuro.com if you're curious to see who and where is publishing. But it's really fantastic to see, where Pipsqueak is being used now. And so with that, I kind of want to focus in on a question that comes up quite a bit, and that's really, what is the difference between these two versions of Pipsqueak that are available? Generally we have Pipsqueak AI on our website, and this is really what we're focused on. But we've continued to make Pipsqueak Basic available. If you download a trial for Pipsqueak Basic, you're also going to get to use Pipsqueak AI for a couple of weeks, so you get to see how that works for you. But really our difference is coming down to the type of cell detection that these two programs can, or that Pipsqueak can do. Our foundation is that we can do image analysis. You can bring in confocal, you can bring in brightfield, you can bring in fluorescence, and we can quantify the cells in those images. We can do co-localization analysis, And generally, we've made Pipsqueak open source in terms of how we're quantifying images in ImageJ. I think this is really important, and this is our kind of fundamental foundation. But how we detect those cells is what differentiates Pipsqueak Basic from Pipsqueak AI. And so in Pipsqueak Basic, you're going to be using a lot of the thresholding capabilities in ImageJ that's really designed to see how many bright pixels are in a cluster, and if that's kind of cellularly shaped. And this is really fantastic. And there was a lot of people that this works really well for. This is what I used my entire postdoc fellowship period. And so you can go a long ways with thresholding. But where I found it wasn't working as well for me is in a lot of my WFA analysis, or if I had background staining in my images. And so that's why we developed AI. And so what's available to you in Pipsqueak AI is our AI-based detection. Now you'll see custom AI development, and then some other additional features on top of that that rely on our AI, such as tiled image analysis. So if you tile a number of small images into a wide frame, Pipsqueak is able to process those very easily or very well. And also what you'll see today is batch image processing. And we think this is a great addition. And so with that, I just want to kind of show you the difference between these different methodologies to detect cells. So with thresholding, what we see is that Pipsqueak does a really good job of finding a lot of these clusters, but it tends to fail when we have multiple cells overlapping, or sometimes a faint cell, or if you have maybe a white matter track going through your image. These are all conditions that are just really difficult for image thresholding to work through. And so this is the reason that we developed our AI. And so now what you can see with our AI detection capabilities is that really it's looking for similarities to previous examples that it's seen. And this is definitely a more robust detection method compared to thresholding. And so here's an example of where I see my thresholding fail a number of times, or quite often. And that's, with these WFA cells, I've got a lot of background. These, perineural nets, these perineural nets are, sort of diffuse and hard to define where they end a lot of times. And so, thresholding isn't great for that. But we developed millions of examples to train our AI with. And when we did that, I can run that exact same image through. And this is the stock output from our WFA model. And you can see just how much better that is than thresholding. So, this is where I think we really see the benefit of the AI. And now what I'm going to show you today is we can continue to improve that AI, because I know that, a lot of the perineural nets that I stain aren't necessarily what your perineural nets look like. And so what we've developed is a way to transfer our AI models over to what you might want to be detecting, or what you might want to be detecting accurately. And so I'm going to show you this Parvell-Beumann model as an example of that. This is one that's generally available in Pipsqueak. You'll see when you open up Pipsqueak. And so here, this is again an untouched image. This is Parvell-Beumann staining. And I analyzed this image with the Parvell-Beumann model. And so, that looks great. I think this is exactly what I would want to see for detection in this image. And so I like it. Now, what we did with this Parvell-Beumann model is we wanted to see how well it would work on another cell type. And so this might be a similar situation to what you have coming in. So we used our Parv model, and we tried to analyze a CFOS stained image. And so, this is encouraging, but it's not great. If I were to get these results back after submitting my CFOS image, I'd feel like I still had a lot of work to do. I'd probably have to go do a lot of manual annotation. And the reason for that is that, those Parv cells, although round and, brighter than the background, actually look quite a bit different than the CFOS staining here. And so, I want to improve this. And the experiment that we did at Rewire, and really what's, led to a lot of our custom model development, is we hand annotated, just a couple images. I think we actually only had to do 10 of these CFOS images. And we took those annotations and we showed them to our AI and continued to train on top of our Parvell-Beumann model. And when we did that, our CFOS model became very accurate for CFOS. And so this is exactly the sort of customization or progression that we're now enabling you to do. So if you do, just a handful of images like this, we can potentially improve the accuracy for your stain coming in significantly. Now, we only had to do, around 10 for this, because these are, great images, just hardly any background. CFOS is a pretty easy stain to see, but that's not always going to be the case. We'd like to say, ideally, you're going to be doing, maybe 30 images. You're annotating 30 images and submitting those, and we'll be able to get to an accurate point for you. But compared back to what you'd be doing by hand to train your own model from scratch, this is nothing. To train a model from scratch is requiring hundreds to thousands of images, and so Pipsqueak is really taking a huge burden off your shoulders just to speed up the development of these AI models. And so, that's what we see. This is some of the data that we've started to publish, and I'm not going to spend too much time on this, but we found that our models can be extremely accurate, given enough good examples. So, these are some of the four most common that we have in there, and I'll show you that we have a number more now. But WFA, parvalbumin, CFOS, and 8-oxo-DG are all extremely accurate, and most importantly, more accurate than analysis done by hand. And so that's where we really think there's a big benefit to this program. And so with that, unless you have any questions at this point, just on kind of the general premise of Pipsqueak, I'm gonna dive into some of the demos, because this is probably the most exciting part of what I have to show you today.
No questions.
So, oh, sorry, Christy.
I was just saying, no questions so far.
All right, great. So, what I've got here is Pipsqueak opened up. And you can see that our menu looks a little bit different than what you might have on your Pipsqueak right now. And that's just because this is, oh, well, wrong version number there, but this is the new version of Pipsqueak, version 5.6. And so the first thing that I'm going to do is I'm going to go into my single label analysis. And right away, if you've used Pipsqueak before, you can see that there's a new option here. So typically, we've seen analyze and we've seen some, but now we've got a batch analysis feature. And so I want to show you how that works real fast, because one of the things that's happening is we're adding a lot of new capabilities, a lot of new options. And so we want a way to really consolidate and streamline image processing. And that's what we're doing with batch. So when I open up batch processing, I can first off just select a folder. And this is really kind of the key to batch processing, is that I'm going to put all of the images that I want to process into a single folder. And so, oops, in this case, you know what, I've already gone through this one, so I'm just going to move all of my files all the images that I want to analyze into a new folder like that. There it is. Great. And so now when I'm over here in Pipsqueak, I'm just going to select that folder. And in this case, these are all WFA images. And I've only got 3 examples so that we're not sitting here watching this process all morning, but this will give you a good idea of how it looks. And so I've got my folder, and now I'm going to select a model. And this is the other really exciting thing that I want to show you today is when you open up your model library in Pipsqueak, you've probably seen these 4 models for a long time, but since then we've gone through and we've trained a number more. And so now you can see some more general models that we think are really good starting points for some of the cells that you might be bringing in. For example, a cytoplasm stained image, might be best analyzed here. And this would be, a great starting point for your custom model. And then down below, I can see a couple new things that are probably even more exciting. One is a model that's trained by another Pipsqueak user. So here, this is an 8-oxo model that was trained by Grant. So these are, maybe just a little bit more specific for Grant's images. And then I can see 2 of the models that I've already completed that are, custom for my analysis. So I've got a WFA and an 8-oxo-DG, and I'll show you how we get to that point here in just a minute. But for now, let's just choose, this vanilla WFA model, and we'll get going here. So really what you're going to see in batch processing is something very similar to what you expect in single label analysis. And that is that Pipsqueak is just going to run through the identification of these cells, but now it's doing it all by itself. So I'm going to sit back. I might go get lunch now that I've started it, and it's just going to find all the cells and all the images that I threw at it. So that's great. And now that it's done with the detection, so it did 3 images like in 10 seconds there. Now that it's done, it's giving me a chance to go back and double check the cells that we've detected, because I think this is a very important part of image analysis. And so here I'm going to say, okay, and now it's going to show me all my images, and it's going to do the background subtraction and quantification that you're used to in Pipsqueak. So here again, I have a chance to tweak things if I want. I can manually move around ROIs if I wanted it to be maybe off to the side here, whatever. And if I like it, I can say I'm finished. And so great, I'm just going to do that for all 3 of these images. And with any luck, this is going to be a very quick step for you, just to say, okay, okay, okay. Another thing that you're noticing here maybe is that there are some new options in our menu, and I'm going to come back and point these out a little bit more in the end. But you can see that we can start to add these images to custom models that we want to develop. So great, that looks like it did our batch processing pretty quickly. No problems there. There is one additional new feature here that I want to point out, and I'm just going to show this really quick before we move into the custom stuff. But one of the things that we've enabled is to analyze images that have both bright and dark backgrounds. With both threshold analysis and with AI analysis or detection, one of the things that becomes difficult is looking at cells that might be stained with like a more typical IHC process rather than immunofluorescence. Because we might be seeing that the cell is dark on a bright background, and so that can be difficult for AI. So let's see. I'm going to go back, and here I've got an example image here that I think has that was just an IHC stained image. And so yeah, great. We're seeing that these are dark perineural nets on a light background. And so I'm gonna skip through some of these settings. These are things that we've really dove into in some of our other webinars. So if you have questions about how to get an image set up, what some settings mean, those are things that you can find on the website now. So I'm just going to click through it. And great, now I've got my background subtraction. And this is really where I have the option to change things. This is a new option down at the bottom. Do I have a dark background or do I have a light background? In this case, I have a bright background. And so I'm going to click that. I'm going to use my automatic background subtraction. And I've talked about this feature before, and I'm not going to go into it today, but how to limit the analysis to a specific region in an image. So great, I've got that. And boom, what you've seen is that Pipsqueak just inverted that image. So the pixel intensities are still representative of what they used to be. It's just inverted it. And now this is able to process through the AI much better than it would with a bright background. Oh, great. And I see a question here for batch processing. Is it possible to go in and select ROIs? Yeah, it is. Absolutely. And so this is obviously not the same batch processing window, but it's very similar. So anytime that Pipsqueak does detection of cells, you end up with a chance to see that detection, see the image, and see those ROIs overlaid on the image. And so this is an opportunity for you to always go in and modify the ROIs, or even load a previous ROI set. Maybe you've done this analysis once before and you want to load in those ROIs instead. Or reprocess maybe with a new model. You want to try out your custom model instead. But yeah, this is a chance for you to do that. And actually, one of the other good questions that comes up quite often is, why do I see 2 versions of my image? This is basically showing you your raw image here. This is not background subtracted, and over here I have background subtracted. This is something that we're probably going to make a little bit more streamlined in the future, but right now you can see both. And sometimes I like to see one versus the other. In this case, if I'm going to go through and look for these WFA cells, I don't think my background subtracted image is actually the one that I want to look at, because some of these WFA cells become pretty dim. So, I tend to look over here at my original image because, when I look at those WFA cells, or the perineural nets, I keep calling them WFA cells. When I look at them, it's much easier to see. And so typically when I'm going through and manually modifying the ROIs, I like to toggle the show ROIs feature on and off on the ROI manager, because that allows me to really look at what's happening here in the image and say, like, okay, there are definitely 2 perineural nets there, 2 perineural nets there. So did Pipsqueak pick those up? Oops, there we go. And so, yep, it got those two. Yep, it got those two. That's fantastic. Oh, but what's this one up here? It looks like it's detected well, maybe that's another one there, although I might argue that that's just some processes coming off that one. So, here's maybe an example where I would just edit this a little bit. I'd delete that ROI and then I'll take it as is.
Got a couple more questions about batch processing, John.
Great. So what if you have some images for batch analysis that you already have ROIs for? Could you load them in the corresponding ROIs? Yes, that's exactly right. So great question. And I'll show you that here now. Again, this is not the batch process, but well, let's go back to batch process because maybe that would be better. So great, I've finished that analysis and I've got my main menu back. So let's go to single label and we'll do batch here, and we can find those batch images again. And there they are. Okay, so we've got our batch images. Let's do WFA again. And actually, this is a good example because now I already have I have ROIs that have been previously generated for all three of these images. So Pipsqueak's going to go through here again, and just 10 seconds or so, we'll get through these images, and we'll load in some previous ROIs. So can one use AI to measure intensity and number from a brightfield image? Yeah, Shafali, that is a fantastic question. You're exactly right that we can certainly detect cells in both brightfield or fluorescence or confocal images. What it really comes down to after we detected it is, is that a staining that you feel comfortable quantifying intensity for? And, I don't think that I can make a blanket statement that it's always appropriate to measure intensity. Certainly with techniques like DAPI or not DAPI, DAB staining, I don't think measuring intensity is probably appropriate, but, I'm not an expert in DAB. So I think it really comes down to, the type of stain that you're putting onto the image and whether or not that's a good one to use. However, if the staining intensity is relative to, the protein level in that image, then I think intensity quantification is great. Either way, Pipsqueak should be able to identify and count those cells. And so here, great, we've gone through batch processing. We've got one of our images here. And so I'm going to load in an ROI set. And when I do that, I can just select that button. And then what it does is it opens up another file browser for me to choose an ROI set. So let's see, I don't know what image that is, but let's just choose this one here, or let's choose a different one. And so when I go into the folder where I'm expecting to find my ROIs, I can actually see the archived version that I previously analyzed. So when Pipsqueak goes through, rather than, saving over your old data, it just archives it so that you always have a copy. And so in this case, I can select that archived copy, say open, and boom, it placed it on there. Obviously, these are not the right ROIs for this image, so it doesn't look right, but that's exactly how that works. One of the things that is upcoming, and I've already had someone ask for, is can we do this for co-localization or for multi-label analysis? Can we do batch processing for multi-label? And the short answer is not yet, but the way that you can do it currently is to analyze each one of those channels, maybe in a separate batch workflow. So do, parv and then do WFA, and then come back and we're able to compile those images and do the co-localization just in one step. Currently, that means maybe just opening it up, loading in those ROIs through a double label analysis and going through co-localization. But that is a feature that we have already written and we'll deploy here soon, so you can just compile some images and get through a batch process co-localization quickly. Great. So question here, Pipsqueak can nicely detect cells, but can it trace the cells that have complex shapes accurately? That's a great question. And how does it calculate intensity? With ROIs, circles or ovals, would it introduce error if the cells have irregular shapes? Yeah, that's a really good question. So one of the things that we adopted in the Sorg lab when looking at our ROIs is that we subtract out the background so that the space around a cell in an ROI is typically set to a, a non-value here. So if you look at my cursor going around this cell, and then you watch up here in my ImageJ browser, you can see that a lot of the pixels around that cell are actually set to nan. And nan just means not a number. That means that they've been subtracted out. And what that does is it removes those pixels from the intensity calculation. They don't contribute to the mean. And so in this case, we feel that we can use, an expanded, a circular, or a square rectangle ROI around a cell and still come up with a shape that's fairly that still represents the intensity of that cell. However, we can also go in and manually draw a shape that better represents that cell. And so in that case, in the case of this WFA cell, or let's look at the non-subtracted image because it's a little bit easier to see. I can go in and I can choose this freehand selection. And, in this case, I might want to draw my ROI, to be something a little bit more like that. Great. I can save that, and, that will be one of my ROIs. You're also able to do that form-fitting ROI through our threshold detection. And this is something that's not available yet, but we are working very hard on enabling, is that form-fitting ROI is going to be available through the AI analysis. And basically, we're just adopting a new AI technique there. Instead of doing object detection, we're going to be using some image or instance segmentation technology to really hone in on just the bounds of that cell. And so that should really answer that question. Until then, I think thresholding or drawing it is a great method. So cool. If there are no other questions about this, I'm actually just going to cancel out of this guy here, and let's go over and I got to close all my windows. That's the fun thing about
one new question.
One new question. Yeah, great. If there are two cell types marked with the same fluorophore, but the user is only interested in counting one or the other independently, is it possible to train the AI so that it distinguishes between the two types? Great question. And how sensitive would it be to the difference? And so what we're deploying right now is the ability to look at one channel at a time. So in my oops, where's my cursor? In my co-localized images, I tend to so here's a triple label analysis or triple label image set, and what I tend to do is export each one of those channels independently, and then also export a merge channel. And so, it's a little bit more straightforward for the AI to go through and look at, this WFA image, or to look at this PARV image independently. But we do have the ability to merge those two models together, and that's something else that we're working on currently. So right now, all of our AI models are single class. We're looking forward to enabling multi-class model generation. And it's fairly straightforward to do. We've just really kind of honed in on this use right now, since a lot of confocal microscopy will show you these channels independently. One of the other things that I think is pretty exciting about that is, we can start to take a merged channel like this, maybe, analyze the WFA and the PARV at the same time, and get all those results all at once. So yeah, let me know if that answers your question, or if I misunderstood it. But
he seems like he's asking about two different types of cells with the same fluorophore, which would be a bit different, I think, John.
Oh, right. I'm sorry. Yeah. So let's see. What's a good example? Well, CFOS is kind of an example of that, or DAPI, difference in morphology. Yeah. I think the best answer for that is that our AI models are really tuned to find what they've been shown as examples. And so we can see some crossover. I think I showed you the example of CFOS crossing over, or PARV crossing over to CFOS. And so the morphological difference here between these two stains was enough to really decrease the accuracy of that model. But that varies. It's going to depend on what's been trained in it. So if you want to come in with a stain, DAPI is maybe a good example, where you can kind of see some differences in cells. But if you come in and you want to train a DAPI model that's got two really distinct morphologies, putting those data in the beginning will enable it to detect them in the end. But we need to show it those examples, and a lot of times it's just a little bit of trial and error at first to see if it works. If it does, that's great, because I think the idea here is if the AI can detect cells at an accurate level for you, then that's perfect. So yeah, and if I think if that doesn't answer your question, I'd be happy to talk with you more after, because I'd be really curious to make sure that we can hit that mark for you.
I think once we get the demo on how to train your own model, that might somewhat answer the question too.
Well, good segue. Yeah. Excellent.
I did that on purpose.
So yeah, let's take a look at that here, and then we can spend any more time on questions and wrap it up after. But yeah, custom AI generation is really neat. We spend a lot of our time at Rewire doing just this. We're developing annotations and training models, but now it's available for you too. And so let's see here. I'm gonna just look for the image that I want to analyze. And oops. Great. And so yeah, so there's two main ways that that we can start to develop our own models. And the first is probably the most straightforward. Just go to this create model button. And essentially what this is doing is giving us an interface to now hone in on where we are in our workflow. So to generate a model, I need to do some annotations, and then I need to submit it for training. And so the first thing on here is add images and ROIs to a custom model data set. Great, that's what I want to do today. The rest of what's on here is really just talking about the quality of the data that we're putting in and the number of images that we're doing. I'll get to this kind of with some examples in the end, but the way to think of it is garbage in, garbage out. Whatever you show the AI is really what the AI is going to learn. And so here I'm gonna open a couple images and just try to give it the best example possible. And we've tried to make that as easy for you as possible. So what I can do is I'm gonna start a new model here. I'm gonna give it a name, and then I can choose where I want to begin. So let's say in this case, I want to begin with that WFA model because I just want to tweak it slightly. So I'm going to select that, and then let's see, I'm going to go in and I'm going to find where are we? I'm going to find an image to analyze. Let's go over to one of those triple label data sets. There we go. So here's a WFA image. So I've got this WFA starting point. I can tune my detection sensitivity and all that, but we can do that again here in a few minutes. The other setting down here is that that tiled image analysis, and I've gone into this in a previous webinar. So if you're curious about how to analyze tiled images, check out I think webinar 2, and you can change those settings down there. Super great. So starting a new model. If I'm starting a new model, the first thing Pipsqueak is going to ask me is to give it a name. So in this case, let's do John's WFA, and then what's today? Oops, no caps lock. Today is 04/27/21. Great. That's just what I'm going to call it. So I've done that, and now it's going to remember that. It's going to actually be saved in a custom model library that I have in Pipsqueak, and I'll be able to continue adding images to that. So here's our WFA detection coming back from the stock AI model, the stock WFA model. And now it's giving me a chance to, to tweak it. So here again, where's my ROI manager? I'm gonna toggle through these and, look at where I wanna change things. I think that there's maybe like one more little perineural net there. And yeah, I don't know, let's say I don't like that one. So I'm gonna delete that one. Great, so I've done that. Going back to my main menu here, I'm going to say that these are complete. I like it. And I'm going to say, okay. And so what that's done is it's now taken that ROI set, that image, and stored it in a special folder for you so that all the other images and ROI sets that you give this name, John's WFA 427, are going to be in that little folder together. So that means when you're training a custom model, if you are looking at multiple different channels, you want to make sure that they go into the right buckets. So if I do another parv image now, or if I do a parv image, I don't want to put it into the same bucket as my WFA image, because those would those would work against each other in terms of training accuracy. So great, let's say I want to add another one to that. I'm going to come back to this menu. I'm going to say add another one. And now what I'm going to see is, oh, here's that model that I just started training. That's now in my little library. That's really nice. So great, I've got all of that. Let's say I did it. And we can go back to our main menu here. Oops, that's not where I want to be. And when I get back to the main menu, now let's say I've done those 30 images or so. I'm ready to submit this. I want it to be trained. And so all I have to do is from my custom model button, I'm going to go submit data set. And when I submit the data set, I have an option pop up here that shows me all of the models that I have in my local custom library. So in this case, well, here's this one that says John's WFA in training. That's one that I submitted yesterday. So I had already done those 30 images and I submitted it. And when I did it, it transferred over to in training. And so here, let's do it today for this new one. I'm going to do that. I'll get a notification that it has been submitted. Great. And at this point, it's taken us about 3 to 4 days to turn these around and get it back to you. Once we do, what you'll see is it now shows up in your custom model library. And so here, I've got to do, yeah. And so, when we submit it, you'll be able to just come back, go to your, any one of your analysis windows, and that's going to be what shows up down here in your custom models. So you'll be able to select John's WFA. Now, if we go back and look at my models in training again, oh yeah, now we can see that one that I just created. So 427 is now in training. So that's pretty cool. It's shown up there, and I know where it is in the process. So outside of that button, we recognize that you're not always going to be sitting down and wanting to just train a model. A lot of times when I'm doing analysis, I am halfway through a WFA image and I'm thinking like, oh gosh, I wish the model, was able to detect that a little bit better, or any one of these markers. And so let's actually look at a triple label analysis. This will be kind of neat. And so I'm gonna open this up. I'm gonna choose all the images that I want to analyze here. Here's a nice little feature that I'll show because I think it's sometimes overlooked. So here I found my images. Here's another triple label set. Here's those 3 channels and a merged image. So what I can do is I can just drag these straight over. So I know that this image is my parv image. I'm going to do parv first. I know that this image is my WFA. I'm going to do that first or second. And then there's my 8-oxo. Down below, I have this option to add a merged image. This is something that's no longer required. So if you don't have a merged channel come off of your fluorescence microscopy or something, whatever your fluorescence scope, you don't have to add it. It will actually just create one for you. I'm going to add it here because I think they're pretty and they're fun to look at. So let's throw that in, and we're going to go through analysis. So here's my my parv image. Oh, there's my WFA image. And there's the merge. Great. So we're getting it all set up. Again, some of those custom settings, you can add in info that will help you sort your data in the end. And here's a chance for us to just double check the different images that we're opening here. And great, I'm just going to do this automatic background subtraction. And so here's my parv image. Great, let's process the parv. And when this comes back, what we will see is an opportunity for us to add these to a custom model. So I don't want to modify these. I think they're great. Maybe I already modified them, but I do want to add them to a custom model. So here again, I've got my custom model library. I don't really see, well, okay, in this case, maybe I do want to do it for the parv. I'm going to add it to that one. Excellent. And it's been added, and now we're going to move on to the WFA image. So here's the WFA image. Again, I'm just going to do the standard automatic background subtraction. We're going to process with the standard WFA model. And when this comes back, well, now I do want to add this to another model, to another custom model, but I don't have a WFA model that's not in training. So I need to start a new one. So I'm going to go start a new one. Let's say I've also already done all my annotation. Great. So here I'm just going to give it a new name. So basically what you can see here is that we're able to do that custom model generation really from wherever you are in the process. And I think that's, something that's important to just making this as smooth and as easy as possible. And then lastly, here's my WFA. So we'll just go through that one really quick too. Or not WFA, this is my 8-oxo. So cool. These are always amazing to see pop up because there are just so many cells in it. It's always really encouraging just to see the work that I don't have to do. To give you an idea of the amount of annotation that went into just this model alone, I think we had nearly 1,000 images, each one of these with like these 500 or so cells on it. And processing or annotating each one of these images, I think takes about an hour by hand. So there's a lot of work that goes into those 8-oxo models. So, all right, I'm going to say it's done. But here's one interesting thing. Now it's remember that I already put it in a WFA model. And in this case, I don't want to put my 8-oxo into the WFA model. So I'm just going to go back and select no here. And basically now this image just isn't being added to any custom. It's just being quantified, and I've got it there in my results. So from there, there are some new features that have shown up recently in the co-localization analysis, and I'll just show this real fast since we're here. But one of the things that I think is important is that our co-localization is a little bit more attuned to looking at the differences between the data sets. And so here, this is not the right cell. I can tell that that parv cell is not the same as this perineural net. So I'm going to go in and I'm going to just maybe select that parv cell. And here I can see now this is the frozen image. This is what I was shown previously. So I have an idea, I have a reference to look at. And here's the image, the parv image that I can manipulate. So I'm going to delete that ROI because that's not the right perineural net. And I'll say, yes, everything's good. And now what's going to happen, this is just slightly different than how co-localization used to work. It is actually going back through and reanalyzing this for co-localization. So what you'll notice here is that perineural net ROI is now absent. It's figured it out that is not one that I wanted to have. And so great, it's finished that up. And I'm going to accept it. And so rather than watching all the rest of the triple label co-localization, let's switch over to answer any other questions you have. And I'm happy to go through any other parts of the customization process that you have that you want to see. I do like seeing how many overlaps with 8-oxo there are in some of these images. It's always fun to just see it process to churn through those. So yeah, let's see any other questions on co-localization. If not, let's take a look at just some of the examples of what really is going to make a good custom model and where you can run into problems. So I've already talked a little bit about this. So generally, training a model takes just thousands and thousands of annotations or images to train a good deep learning object detection model. So we've done a lot of the heavy lifting for you because now Pipsqueak is able to do this in just around 30 images or so. And as we saw with that CFOS model, we can get down to even fewer, and it's pretty exciting. But one of the things that you have to watch out for is examples or times where maybe you're not annotating all the images in that, or all the cells in that image. Here's an example of this is an image that's been sectioned down, so we're just looking at a stripe through it, but there's a lot of maybe parv here that wasn't annotated. And so when we show the AI that during training, it basically gives it a bunch of false negatives where these are cells that you probably want to detect, but in this case they would not be contributing to that learning, and so they would detract from the accuracy. Whereas this example here is showing all of those parv cells being annotated with ROIs, and so that's great. So kind of some general rules on what you might be looking for is what I said earlier: garbage in, garbage out, and that false positives and false negatives matter. So if you've gone through and annotated an image like this where there again just thousands or hundreds of cells here, you really want to make sure that you've got all of the cells that you'd be looking for. So I think this might be like a CFOS image here or something like that. And so, this is one that we tried to do the heavy lifting for you. We've trained a CFOS model. We've trained or we have a DAPI model that's about to be released. Since, these are such ubiquitous stain types or cells that are stained with that, we tried to go through and do all that annotation for you. And then kind of the last thing here is not super easy to see, I'm sure, especially over Zoom. But generally, when I am annotating an image, when I'm annotating a cell, so I'm going to just cancel out of all this, and I'm going to show you a good example of an annotation. So here, let's pull up a let's pull up a merged image since they're better to look at. And if I were to go in and try to put an ROI around maybe this perineural net in green here, I really have a couple options. One is to draw that ROI really tight, against that cell. And I could even go into the point where I was not showing, where I was not including the borders of the cell. But what I'm looking for, a good annotation here, is one that actually encompasses a little bit of background around the cell. So here, I'm gonna try to make sure that I've included a little dendrites that I want in my detection. But you can see there's a lot of space around that perineural net, and that's great. Those are examples for the AI that show kind of the border of a positive and a negative. And that really helps contribute to an accurate model. So that's that's an annotation that I would take and move forward with. So with that, I think the rule of thumb is really to go through and accept annotations that you would want to see in your analysis anyway, because what you contribute in is what you're going to be getting back. But it's pretty exciting to see just how easy it is to really move this AI over to detecting a cell type better for you. And so I'm really excited to see what you can do with it. As always, we're here to help. I think one of the things that we really pride ourselves on at Rewire is just being available to answer any questions or fix any problems that come up. And so as you're going through this, don't hesitate to send me an email at john@rewireneuro.com, or reach out to our support email, which is just support@rewireneuro.com. And so through one of those, we'll get back to you, and we'll be excited to help you get going on this, because it's a neat capability to be able to really detect things more accurately. So with that, if you don't have any other questions, we can wrap it up here, and you can have a little bit of your hour back. Thank you so much for coming and being here with us today. Oops, two more real fast. Plans to develop an OTX model. I do. Well, actually, coming out of the Sorg lab, I think we have some OTX annotations that can go into a model. And so Shefali, if you have more OTX annotations to contribute to that, we might be able to merge them together and create something that's even more accurate. So let's talk offline about that, because that'd be really fun. And Connor, when you're analyzing images and adding it to your custom model at the same time, does it make two separate folders? Yeah, great question, Connor. So that's one of the really important things to think about it. Here in this triple label example, I was adding some images to WFA, I was adding some images to PARV, but I really need to make sure that I'm selecting the right folder to put it into. If I put my PARV image into my WFA folder, those are not going to be contributing to the accuracy. They're going to work against each other. So as I go through, just choose the one you want it to be in, as simple as that, and it'll be set to train. So thank you so much, everyone. It's really great to see you all here, and looking forward to seeing what you can do with this.
I'm gonna go ahead and launch the poll.
Oh, that's right. Yeah.
We'll still have more questions. Let us know. Here we go. Did that work? Okay.
Yeah. Wonderful. Yeah, well, thank you again. We will put this webinar up online here as soon as possible, probably within the end of the week. And we'll try to break our content up a little bit more. So, as you're going through this webinar or some of our previous webinars, you might be able to just hone in on that one little section that you want to rewatch. So yeah, just check the website for that, rewireneuro.com. And again, thank you for being here, and feel free to reach out to me with any questions that you have. So thanks so much, everyone.